Dr. Keloth, shall we start?
Or
Can you unmute your mic? Yeah, fine. So let's start.
Yeah, you can. My screen is still shared.
Yes, it's in share. Yeah, it's getting shared. No problem.
Okay, so I should keep sharing.
Okay, sure. Yeah. So
Okay.
welcome everyone and hello to the Progress and Prospects in Biology YouTube channel. Today we are having our 22nd talk, and today we have with us Dr. Rajmohna Keloth from Zoological Survey of India to deliver today's talk. As you are all aware that we have transitioned to this new format where the talks are being broadcasted live only on our YouTube channel and also on our Facebook page. So before we begin, I would like to introduce Dr. Keloth to all of you. Dr. Rajmohna Keloth did a PhD from University of Calicut under guidance of Professor T.C. Narendran. She then joined as a research associate at CSIR. Then she joined Zoological Survey of India, and currently she is scientist E over there, and she has been awarded High Performance Award of the Zoological Survey of India for the year 2011 and 2012. She serves on the editorial board of many international journals of international repute. She has delivered many invited talks in national and international meetings. She has described 145 species of insects, and which are well documented and published. And she also found new record of 17 species of fauna in India. And she is a very renowned scientist in the field of taxonomy in our country. And she will be delivering our today's talk, which is entitled Exploring the Unknown, a taxonomist's perspective. So, to all the participants, please post your questions by commenting in the chat box. We will try getting them answered towards the end. So, with that, over to you, Dr. Keloth.
Thank you very much. Good evening to all. I'm really glad for this opportunity to interact with you. Thank you, Professor Ina Ray Banerjee, for inviting me to this event. I'm also thanking Director Zoological Survey of India for this great opportunity. So I have designed this talk, Exploring the Unknown, a Taxonomist Perspective, especially for the student community. I've tried my best to share my experiences over the past more than 26 years as a taxonomist, insect taxonomist in particular. The challenges involved in doing the taxonomy, also the joy of describing new taxa to science. One thing I would like to add, you all will be smiling to your friends, to your relatives, but we taxonomists smile at our specimens too when they surprise us from under the microscope lens. So that much is our attachment with the specimens. I would also like to add that I'm a traditional classical taxonomist, but I'm very much open to the new technologies and interventions of molecular taxonomy. So doing taxonomy is not the same in the case of all groups. Being experienced with insects, all my examples and explanations will be based on insect taxonomy today. So to demonstrate the difference between a highly diverse and a little studied taxa, I'll be quoting examples from parasitic Hymenoptera, my principal group of research. I started my research in parasitic Hymenoptera of India. Later, I also started doing some taxonomy in the case of termites of India. So the two groups are entirely different in the sense one is well known, very less diverse in the case of termites, whereas the parasitic Hymenoptera is highly diverse and little known. So that we can just analyze the experience between doing the two types of taxonomy. Okay, so let's go to the talk. The first part of wisdom is getting the names right. This I have quoted specifically for the binomial nomenclature. This naming, international system of naming with a genus name and a species name was to ensure a universal acceptance of names, and introduced by Linnaeus, father of taxonomy. We will be mentioning Linnaeus too many times in this talk. And the 10th edition of Systema Naturae, you all will be aware, which got published in 1758, is considered as the beginning of zoological nomenclature. See, diversity is the main attraction of life on Earth. How many species are there on Earth? This is a fundamental question. Everybody can ask, but it's very difficult to answer. There were many attempts by several persons to try to decipher a somewhat accurate figure. But there were a lot of controversies based on the methodology involved and all that. Mora et al. 2011 is a widely accepted paper where she describes 8.7 million as the figure of eukaryotic species globally. 7.77 million with a standard error, it's written there, is Animalia. 2.6 million are insects. See, the next word, next line is very important. 1.2 million are named. What does it mean? Out of 8.7, we know only about 1.2 million. That means a lot still remain to be discovered. 2.2 million of them are marine, then rest are terrestrial. So 86% of the terrestrial species and 91% of marine species still await description. That is the huge task in front of taxonomist. So this is a 1.2 million is a figure in 2011. Now it is almost 1.5, the total number of described species. So this method was based on validating the higher taxon approach. Like a pattern is there for predicting the higher classification of species. So in that particular pattern, the number of species can also be predicted. This paper is available online, and those who are interested can go through it in detail. 2011 onwards, there was a series of publications from Zhang regarding the described diversity of Animalia. 15,52,319 species of animals in 40 phyla is according to the new evolutionary classification. Phylum Arthropoda, comprising of insects, spiders, this prawns, crabs. This group is the major share of it, with 80% of the total. Insecta coming to 66% of the total. So this is the described diversity. 95% of entire living organisms are invertebrates globally. Among that invertebrates, see, if you just see this particular pie, 82.82% are arthropods, like what we just mentioned earlier. Rest of them are all very small slice of pie here. So Desai has been bringing out animal discoveries for the past 10 years. It gives you that every year how many new species are added, how many new reports to India are added, and overall summary of the described species of that particular year. So in 2019, as per the report, in India we have 98,616 animal species, and if you add the protista to it, it exceeds 1 lakh. Also, in the animal discoveries, there is a comparison of the new species added by ZSI and also by others. Also, new records to India. In the last 10 years, you can see phylum Arthropoda. In that phylum, around 3,411 species were added as new record to India, and almost half of it as new species. So we had just now mentioned about Arthropoda being a very big group in invertebrata. See, the hexapoda of the 4 subphylum, hexapoda is again the largest of the subphylum. Under hexapoda comes insecta. In the class insecta, you can see different insect orders mentioned here. Coleoptera is the largest. Lepidoptera means butterflies and moths. They are the second. Diptera comes third, which are flies. Hymenoptera comes the fourth position. So this is all in nutshell about the huge diversity which we are having, and this data is from 2017, this particular paper. So I'll be concentrating a little bit on Hymenoptera just to explain or to demonstrate how diverse the group is, and in such groups how taxonomy is being done. 13% of all known species of insects are hymenopterans. So hymenopterans means wasps, bees, ants. They are the familiar groups. So again, I'm not going into the detail of the classification, but still there are 2 suborders, Symphyta and Apocrita. You can see here there is no wasp-like constriction between the thorax and the abdomen. They are soft lice, and here just imagine a bee which is having a good constriction between thorax and abdomen. Such groups are called Apocrita, under which comes parasitic Hymenoptera and also the stinging groups, bees and wasps. So Hymenoptera is a big order. We have 27 superfamilies under it, 132 families, and 8,423 currently valid genera. Here comes the next important aspect. More than 75% of Hymenoptera are parasitic Hymenoptera, and we know them very little. So in India, there are about 10,000 plus species of Hymenoptera, and 75% of it means a huge number. So what are they? They are very minute. That is the first property I will say, first character. They are natural enemies of a large group of insects and some arthropods. They control or regulate the population of these insects and arthropods. The adults of parasitic Hymenoptera are free living, but the immature stages are parasitic on their host. There are egg parasitoids, larval parasitoids, pupal or prepupal parasitoids. Means these parasitic Hymenoptera, they lay their eggs inside the eggs or on the larvae or inside the pupae of other insect groups. Even live insects like aphids and scale insects are attacked. So that is how in our ecosystem a balance is maintained between the populations of various insect groups. It is being regulated always. For this property, for this character, uh, parasitic hymenoptera are utilized in biological control in the field of agriculture. We have 13 superfamilies in India. I'm not reading through, but just see here. Chalcidoidea and Ichneumonidoidea. They are the largest groups. In the sense, we have more than 3,000 species in Chalcidoidea and 2,575 species under Ichneumonidoidea. They are again, as I told you, um, egg, larval and pupal parasitoids of a wide group of insects and arthropods. So this is an interesting comparison. Out of the 13 superfamily listed here, the first 9, if you count all the species, it will come less than 150 species. Some of them have only one species in it, some have 10 species, like that. But I should say Diaproidea and Seraphanoidea. I have direct experience in working in these two groups. They are highly diverse and very little explored. So also is Cynipoidea. So if somebody comes forward and uh, takes the task of exploring these groups, I am sure Seraphanoidea will add more than 200-300 species in India. Diaproidea also will add another 100 to 200 species. Cynipoidea also will easily add 100-200 species. So a lot remain to be explored in this particular groups. Megaliroidea is a superfamily. So what is special about it? First of all, it is a very beautiful insect. See the long ovipositor. It is this wasp is parasitizing the larvae of wood-boring insects. It inserts this ovipositor through the bark of the trees and lays its eggs on the larvae of the wood-boring insects inside the trees. That is why it should have this much long ovipositor to explore and find out inside the stems and branches of trees. So this particular superfamily was reported from India only last year. Such things do happen because very less people are working on such groups and the group is very less explored. So reporting a superfamily for the first time is a matter of joy. It was also first report from the Oriental region. This kind of rare discoveries happen only in the case of less studied groups. Family Megaspilidae. This is coming under Seraphanoidea, which I just now mentioned. Again, in just some 3 or 4 years ago, we could report first genus of this particular Conostigmus from India. Again, because see, the world have 323 species. In India, we have only 8 species, means we don't know about them. We could work out little bit, but we are still to publish the major results. Some 22-23 species we could add under this particular family through our work in Kerala. See, this picture is photo is very clear, but the specimen is less than 2 millimeter. You can see the scale given. We always give the scale to have a relative idea of how big the specimen is. It's very easy to identify this particular group. See this particular pterostigma and this curved upwards stigmal vein. So with this combination, it can be very easily identified, this particular family. So also Seraphonidae is another family under the same superfamily. Both of these are hyperparasitoids of aphids, and also some are attacking cecidumid dipteran flies also, gall-making flies. This is the male antenna, this is the female antenna. See, here again the up-curved stigmal vein without that black patch. This is how we distinguish these two families. Again, the world is having 315 species, and we know only about 3 species. We could add through our work in Kerala about 35 species, the new species, and they are in the process of publication. Superfamily Cynipoidea, it's another beautiful group of insects, still little bit bigger to what we saw earlier, but still well under 3 millimeter, 5 millimeter in length. We could record subfamily Fidgetinae for the first time from India. So they are also this hyperparasitoids, and some are directly parasitoids of other insects. So when I say hyperparasitoid, it means they are parasitoids of parasitoids. They are somewhere at the 4th trophic level. And we mentioned about the biological control programs through this parasitic Hymenoptera. So the presence of hyperparasitoids in the field is not ideal for success of biological control, because they attack the parasitoids, and the efficiency of the biocontrol program is lessened. I have included this slide in particular because I was so excited by this discovery. I had read earlier about Foraceae, but never thought that we'll get a chance to encounter or to see it in front of my eyes. As a story, I can tell, but there is no time for such an elaboration. First, one of my student brought this particular bug in this copulating position itself. He traveled for 3 to 4 hours in the train, and he was carrying this particular bug, Cletus bug, hemipteran. This is a heteropteran bug with a small insect sitting on the head. And all this 4 to 5 hour journey, the insect was not shifting its position. Another time, a professor, a lecturer from a nearby college brought to me this particular bug, plataspid bug, Megacopter cribraria. It is a pest. So on it was sitting some 4 small insects. Again, they were not shifting their positions. Even if you keep it for 2 to 3 days, they won't move much. They won't eat, they won't drink, they will just remain as such. And on a grasshopper also we could, this photo was shared with me by Alfred Daniel. These small parasitoid wasps, they used to cling on the body of this particular grasshopper, and they used to feed on the hemolymph here. This is from literature, this photograph. See on the legs, several insects are there. So what is this all about? They are egg parasitoids, and they make use of the host to reach the site of egg laying. They are simply waiting till the host lay their eggs, so that directly at the moment itself they can attack or parasitize the eggs. Poor host, it is carrying the parasitoids all the way it is going, and detrimental to its own next generation. So why this particular phenomenon? One hypothesis is that this particular group of insects, they are able to break that chorion of the insect egg only during the early hours of oviposition. After which, once it hardens, it is difficult for them to insert their ovipositor into the eggs. So that is the main reason why they are phoretic. This is an exciting part where new genera were described under superfamily Platygastroidea and Diaproidea. This Neacerotobius, this is an egg parasitoid of spider. This is a new genus described by me. Then these two come under this particular superfamily, the Diaproidea, and they are pupal parasitoids of dipterans, whereas Anokha, Neodatta, and Chakra, we have the privilege to name them. And as authors, we are immortal in science. So those are joyful moments which is really hard to explain. So these kind of discoveries are not very frequent, not very common. This happens mainly in groups which are less studied. So the challenges involved are also that high while studying such groups, because there is no prior good platform on which you can do the studies. You have to develop, you have to search the literature, you have to contact the experts. From the scratch you are beginning, and you get an insect while going through your collections, and which is not matching to any of the descriptions already available in front of you. It is not fitting into any of the taxonomic keys which are aid to the identification. So there, in that point starts this particular inquiry regarding what this particular organism is. And finally, after going through a lot of literature, all the published information on related groups, you reach a point that they are totally undescribed, and it's not fitting into any of the current species concept. So a new genus has to be erected so that this species can be accommodated. That is how a new genus is born. So that much about the group which is less studied. Now let me tell you something on a group which is very widely studied and having only very less diversity. Termites as a whole in the world have only 3,000 species, and we have 10% nearly of it, that is nearly 300 species in India. And it's a social, highly social organism, and rigid caste system is there. You may have seen in plenty the flying forms which enter during this crepuscular times, near the tube lights and all, near the street lights, they will be flying. So they are the elite, the reproductives, whereas the soldiers and the workers, they remain in the nest. All the morphological studies on this particular group, termites, are mainly based on the soldier caste. So what does it mean? Until and unless you get the soldier, it will be difficult for you to identify even the genera or the species. But though very widely known and highly studied group, a 3-year intense study on the taxonomy of termites of Kerala, that is the Western Ghat part of Kerala, we could bring out one new genus and 14 species new to science. Also, one genus and 2 species as new reports to India, and 20 species as new state records. So we are adding to the knowledge of this particular group. Each species added means species are building blocks of this diversity. So we are getting to know the group better and better each time we get hold of a different species. In the photograph, I have included the most primitive termite in India, and it is confined to Western Himalaya. Only one species under this particular genus we have in India. It's comparatively a big specimen, almost like 5 millimeter plus. It can be up to 8 to 10 millimeter, like 1 centimeter. It lives inside the dead branches of live trees. So it's really hard to find them. So these are some of the new taxa discovered and published in the case of termites. See, when we talk about termites, it is a wrong notion that all are pests. We know termites as pests only. That is our ignorance of facts. Only 12% of the total termites are pests, means they attack trees and cause huge economic damage. Rest of the groups are of great ecological importance. They are mainly soil dwellers. They remain in the soil. They don't attack the wood, particularly live wood. So these, where you are seeing these twisted mandibles and all, these groups are not in the pest category. They are all soil feeding termites. This Glyptotermis teraharite, which was discovered from the rainforest or the evergreen forests of Western Ghats in Malabar Wildlife Sanctuary. It was residing inside a fallen tree. Means the tree was almost in the decaying process. So we were just seeing what all things are there inside, and we got this particular species, and it happened to be new to science. So what are the challenges in termite taxonomy? Though we have the fauna volume of India, excellent publication covering the entire termites, the species, keys are there, good pictures are there, but still, in the case of soil feeding termites, which are of course a good number, we need to work out the gut morphology of the worker caste. In some groups, they don't have soldier caste. Soldierless groups are there, and in some cases, you hardly find a soldier. So in such cases, identification is very difficult, and you have to dissect out the alimentary canal, and near that hind gut, there are certain compartments, and inside the gut you have spines arranged. This is the enteric wall armature, you call it. Spines are arranged in some definite patterns, and based on that, species are identified. Even though a well worked out group, we don't have a global online database or a national database. So it is proposed that an online database is very much needed for good taxonomy to continue, having species descriptions, distribution maps, identification keys, then the cuticular hydrocarbon profiles, also with data on the fungal symbionts and the gut symbionts. Gut symbionts are mostly bacteria in the case of higher termites, and in the case of lower termites, like this, there are, out of the 6 families in India, only one family, Termitidae, which is about 70% of the total diversity. It comes under higher termites because their gut symbiont are mainly bacteria. In the rest of the termites, the 5 families, lower termites, they have flagellates as gut symbionts. So this is the difference between higher and lower termites. So that much about the group-based explanations. Taxonomy, systematics, biogeography, all these are very familiar terms to us. And taxonomy and systematics are often interchangeably used by people, but that is a wrong notion. Taxonomy is not the same as systematics. Taxonomy is the theory and practice of identifying, describing, naming, and classifying organisms, whereas in systematics, the relationships between organisms also is involved. Classification is merely arrangement of organisms into groups. See, if you want to identify an ant, first you have to classify it that it is not a bee. An ant has to be classified as an ant. So identification can follow only after once you classify them somewhere. So this is classification is a step prior to identification. Biogeography is a very interesting field where distribution of species and ecosystems are dealt with. And Alfred Wallace was one of the pioneers who did very notable contributions in the field of distribution of animals, like establishing the zoo geographical realms, the Oriental region, Palearctic region, like that. So the lack of knowledge on how many species exist, that term is generally called Linnaean shortfall, and the inadequate knowledge on the distribution of species is called the Wallacean shortfall. So Linnaean taxonomy is mostly based on morphological characters, and what we do in the case of insects mostly are also Linnaean taxonomy. We study the morphological characters and group them accordingly into species, then also into higher classifications also. When we describe new species, the morphological characters are compared with similar groups and the diagnosis is defined. Same way, the species, the specimens which we study, we are classifying them into the taxonomic hierarchy. So for all this, we are using generally morphology, and this is called morphological characterization. When we are talking about that distribution of species, species are not evenly distributed everywhere. We have 36 biodiversity hotspots all over the world, of which 4 are in India. This is Western Ghats and Sri Lanka, Himalaya, Indo-Burma, and Sundarland. The Nicobar group of islands fall into Sundarland biodiversity hotspot, whereas Andaman fall under the Indo-Burmese biodiversity hotspot. So what is the difference? Here the fauna will be mostly having affinity to the Southeast Asian fauna. In the limited time, I am trying to focus on certain very important concepts in the taxonomy, particularly the classical taxonomy. Type species and the type specimens. The prior principle of typification is one of the major concepts where the name of the taxon to be applied is being explained. It is as per the International Code of Zoological Nomenclature. Again, for maximum stability, there are certain rules which are governed by this convention. How to name a species, what all things are to be considered when naming a species. So all these are coming under the ICZN, International Code for Zoological Nomenclature. When you describe a new species, each and every species should have a name bearing specimen. A physical specimen is tied with a name. That is called the type specimen. And that one specimen which is tied to the name of the species is called the holotype, and it is described in the first publication of that particular species in the paper. Along with the holotype, there will be some other specimens also, which forms the basis of this description, and those are called paratypes. In earlier days, they did not designate one particular species. They used to designate a series of specimens, and those are called syntypes. Later, when somebody is studying that museum collections, they would choose one particular specimen of those syntypes and erect them as a lectotype for the particular species. In case the type is not designated, or in case the type is lost or damaged, the subsequent worker can erect the neotype. This type not being erected and all are not something which is happening in the recent. This is all about the early days prior to the ICZN and like the time of Linnaeus and later workers. So, what is a type species? When you are describing a new genus, you will be erecting a new a species based for which that genus has been erected. So, even if you having two species new under that particular genus, you are free to choose one species out of the two as the type species and one particular specimen as the type specimen of the type species. So this is how a strong root or foundation is built for each species, and such type specimens are of great importance in taxonomy. They are kept in national and international repositories, and it is available for experts to study at any point of time. Nowadays, uh, when the digitizing procedures are very much mm, developed, the museums are putting in their website very clear high resolution pictures of the types, so that you can save a trip personally to the museum and study the types. A small question here. Homo sapiens is a species. We humans, we are species Homo sapiens. So there should be a type for human beings also. Who can be? Who is the type of Homo sapiens? The answer is Linnaeus himself. But it was not erected by Linnaeus. Later, in 1959, Professor William Stern erected the lectotype of Homo sapiens and designated Linnaeus. Just as for some interest only, I shared this information. So all taxabiotic surveys of Zoological Survey of India. Another very interesting episode or a part of taxonomic studies is the surveys you are undertaking. To get the specimens, you have to be in the field. So the field experience is something unique. The terrain can be friendly, the terrain can be unfriendly, there can be many untoward, unexpected incidents happening, but also the joys of the surveys are something which is difficult to explain. You are inside the forest for days together. You are free to breathe the non-polluted air, at least the oxygen, full of oxygen for a long time. You are in the midst of the greenery and you are exploring, you are collecting based on the permission which you got from the forest department. So what is special about this particular two photographs? I always keep these in my presentation. These are one of my favorite because, see, when we go for surveys, we collect specimens from everywhere. It can be aquatic, from water bodies we collect, from terrestrial, like by sweeping, or from the grass, from the soil, even from the canopies, even from the leaves. From everywhere organisms are collected. So likewise, this dead and decayed trees, you will find plenty in the forest. They are in different stages of decomposition. So we are just disturbing that particular big stem or trunk or branch of a tree. You won't imagine what all things, how many groups of organisms will come out of that particular habitat. You will get a lot of termites, you will get a lot of ants, then centipedes, then millipedes, earwigs, spiders, some beetles. These are all common taxa which I told you. And this much big trunk, if you can collect the total fauna coming out of it, and if you can identify them up to the species level, it is worth a doctoral degree for you. But the thing is that it is easily said than done. We don't have that much specialists to identify all the groups which come out of this particular this small log itself. So that much is our diversity, particularly because we are a tropical country. We are blessed with diversity. India is a mega diversity country. Types of surveys are according to what you plan. Faunistic survey, it can be based on political boundary that you are surveying the total fauna of West Bengal, Kerala, like that, or you can select one particular protected area like Corbett Tiger Reserve and do the study there. But you are studying all the fauna over there, even the birds, even the fishes in the water bodies, and you are bringing out a solid document. Taxonomic survey is what, as a hardcore taxonomist, you perform. You are studying only the frogs, or only some insects like only termites, only bees. Then you go to the field only to collect that particular group. So that comes under taxonomic survey. Suppose you want to know the entire fauna of ecosystems, like mangrove ecosystem is there, freshwater ecosystem is there, desert ecosystem is there, then it's ecosystem survey. And suppose the you want to know the status of some particular selected group of animals, maybe coming under the IUCN protected endangered category, like that. The population of such species have to be assessed. So that comes under status survey. So these are the types of publications coming out of it. These are the methods of insect collection. Like pitfall traps are there, sweep nets are there, malaise traps are there, yellow pan traps. All give you different samples. So I am of the opinion that employ all of them for the best representative sample. In the case of termites and ants, we have to use some other kinds of methods like forceps, brush, and trowels. Once you do the collection, next part is preservation. In the field itself, you are preserving it either as dry collection in the case of butterflies or donata and all, whereas in other cases, most of the cases of insects, we are preserving it as wet collection in 70 to 80% alcohol, or if you are interested in the molecular barcoding, then you have to preserve them in 100% alcohol. And you are keeping them in small, based on the size of the specimen, you are keeping in vials and under refrigeration. For taxonomic studies, if the specimen is too small, you are mounting it at the tip of the card, or you are pinning it, you are passing a needle through the thorax, and then you are studying the specimen. For soft-bodied small insects, slide mounting is the option. We don't, as a classical taxonomy or traditional taxonomy, we don't need that many kind of gadgets and all. A very good microscope which can give you good magnification is all what we need. Even a 1 mm insect is magnified in the screen, and see the photographs taken. Each and every part is so clear, the sculpture of the insect is very clear. So this is the series of head front view of these are all 6 different species of a genus called Gryon, and under family Celeonidae, and you can make out how they are different, and this is called morphological characterization. We describe how we feel, and this is how species are differing. See the abdomen like that. There's 6 species of insects. Everywhere there is difference. Even the sculpture, even the total length of this segment. This is called first metasomal segment, second metasomal segment. Here you see how it is different. The color is different. The sculpture is different. The texture is different. The hair pattern is different. So, in short, you should have a very good analytical eye to bring about all this and compile all this information into what you call a taxonomic description. So once the molecular data started entering the traditional taxonomy, things started changing. Even classifications were changed. Then evolutionary theories came into routine animal taxonomy, and the classification itself started changing. When we talk about evolution, the fossils are very much important. Though 2-dimensional fossils are available, mostly in the case of this big rocks and sediments and all. Even soft-bodied insects and soft-bodied even birds are preserved in amber. It is an exudate from a tree. Under stress, the tree secretes that resin, and the insects and different organisms are trapped. And based on the era or the region, we have different kinds of ambers, the Baltic amber, Dominican amber, then the Burmese amber, all are from different ages and tells you about organisms of the past. This is the tree of relationships, phylogeny, which we call it. Common. When we talk about evolution, it is not just change alone. It is a common, and evolution usually is linked with a common ancestor. And it is the descent with modification. After this, the tree got a little bit advanced, and finally, this is the current phylogenetic tree of all life systems on Earth, and the starting point is from the center. Here you have this eukaryote, archaea, and bacteria classification, and the animal clade is here. If you click on this, this is the whole animal clade with vertebrates and invertebrates, and here is the vertebrate clade. If you click on that, this is how the vertebrates in Earth are related. This is made on the basis of ribosomal DNA of about 3,000 species representing the major branches of tree of life. I'm not going into detail. This is about the convergent and divergent characters. Though the water lily, and this is, I took one example, classic example from the plant, because water lotus and lily, you feel that they are closely related, but it's not the thing. When you compare with the sycamore tree, water lotus is more related to sycamore tree than to the water lily. And see this wings of animals. This is of an insect. It's entirely different, though the function is the same. The bird wing is different. The bat wing is different. So they are not homologous organs. The changes in insect classification happened mainly because of the the advent of molecular taxonomy. And one of the main example I can quote is the death of an insect order. Means termites were earlier considered as a separate order called Isoptera, but molecular studies proved that they are related to cockroaches, Blatodia. So now they are under infra-order this Isoptera under Blatodia. This is a primitive termite seen only in Australia, Mastotermus, and they are having very close relationship with the molecular studies have proved with Cryptocercus, another primitive uh, this cockroach seen in North America and some Asian countries. This termite also has ootheca like the cockroach, and that Cryptocercus cockroach is having the gut bacteria for good digestion as the termites. So that is how they linked that they belong to a common ancestor. Some examples from the vertebrate I can quote here. Based on the morphology in 2007, these two species of frogs, Philotus ocalandre and Philotus great, based on the tree dendrogram prepared, they were considered as very closely related species. But later, with the advent of molecular taxonomy, the DNA studies proved that they are not closely related and they are entirely, they are distantly placed in the tree. So this is the example of a phylogenetic tree where common ancestry is the basis of classification. Cryptic species is another very important topic in taxonomy. This is called those species which are genetically isolated but morphologically non-distinguishable. They may look alike, but they are not the same. Such species are called cryptic species. Suppose this is the distribution, you are sampling it, and you are doing the molecular taxonomy, you find some differences here. Then you go back to the specimen. Now you are characterizing some 8 features and you find some differences between species 1 and species 2, and this is how sibling species are tackled. You have to revisit morphology, and it is defined that no two species, biological species are always alike. There will be some minute differences. Only thing is that our taxonomy is not refined enough to find out or to describe that fine differences. When we do the traditional taxonomy and when we do the integrated taxonomy means you are adding molecular or some other ecological or biological features into it. So the traditional taxonomy says in the case of Hanuman langur only 1 species, but integrated taxonomy says 3 species. See, in all the cases, amphibians, reptiles, many examples are given here. The compared to traditional taxonomy, in this integral taxonomy is giving more number of species. So that is mainly because the cryptic diversity is coming out. Another interesting example is of the this Hanuman langur. Earlier, it was thought only 1 species was there in India, and it are coming under Semnopithecus, and the other this Nilgiri langur of the south and this purple-faced langur. They all people thought that it was more related to Trachypithecus, but this molecular studies proved otherwise, and I'm not going into the detail of it. Finally, it was decided that it comprises of 2 distinct species complex and a hybrid species. So, species description based on morphological characterization plus DNA barcoding is now developing as a new formula. Morphology is required for field-based identification. Then single gene sequencing can be done with mitochondrial cytochrome oxidase 1. Why cytochrome oxidase 1? The mutation rate is fast enough to identify closely related species, and the sequence are conserved among conspecifics. So the molecular characterization plus the DNA barcode of that particular, if you do for a new species, that taxonomy is almost stable. So these are the steps for DNA barcoding: extraction, first step, PCR amplification, DNA sequencing. Here ends that DNA barcoding. If you are proceeding to phylogeny, then only this computational biology comes, whether you want to find out the species is of a common ancestry or if it is a monophyletic or paraphyletic or polyphyletic, like that. Only then you will have to proceed to this second part. Nowadays, this DNA barcodes are being used to identify unknown biological material. But there is a word of caution based on the 2019 paper published in PLOS, assessment of bold and gene bank. These are databases of DNA barcodes. What is the accuracy and reliability of the identification of the biological materials in the DNA databases? So just see this graph. Uh, the accuracy is not much. 70% accuracy was seen in gene bank for the genus, and in the bold it was 50% accuracy. For this is for the insect group. In the case of species, in the bold it is hardly 30% accuracy, and in the gene bank it is nearly this 50% accuracy. So when you are identifying, when you are giving a name based on blasting, it should be taken little bit caution. Maybe the situations will change later, but this is the situation now. This is mainly because many species are wrongly identified, and their DNA barcode is just uploaded as such, and that is how you get the wrong combination. We happen to notice this because in the case of termites, we could see a lot of species names which are not reported in India, and the barcodes were available. So the only possibility is that those are wrongly identified species. There is a great taxonomic impediment which is defined as the biological studies suffer due to the shortcomings from the taxonomic point of view. We don't have enough taxonomists. A major portion of global biodiversity is still unnamed. Then describing taxonomy in the traditional method is very slow and tedious. And we need more people to come into the field, and we need more technology also. So, turbo taxonomy is a term which is very popular recently, where they have what we do is just the same, but they have reduced the overall procedure here. The morphological descriptions are not very elaborate, only the diagnosis and very sharp features by which it can be distinguished from its nearest relative is mentioned. High resolution digital imaging which explains those characters, plus the mitochondrial cytochrome oxidase 1 sequence. This combination is enough to describe a species, and in many hyperdiverse taxa, like Ichneumonidae, parasitic hymenoptera. So in the case of certain mollusks also, there are several papers. If you go to the net and you browse this, the turbo taxonomy, hundreds of species are described together using this technique. To accelerate species discoveries, we need some automation. So there are interactive keys. The keys are dichotomous keys. You may be familiar in the textbooks and all, which gives different couplets. And if you run through the key with your specimen, you will reach the correct identification. Once you reach the correct identification, then you go to the species description or the genus description to know what species or what genus it is. But with the computer and the software all available, interactive identification keys have been developed. As a sample, we wanted to experiment how it is all about. So a small key was made for diaphrine genera. It's about 17, 18 genera in this particular group, and it is available in NBAR website. It was a joint venture between ZSI and NBAR. Recently, a wonderful database on Indian Westpid wasps with about 288 species. The fact sheets, you can see what all it contains, morphology, then the checklist, references, everything you want to know in a finger click. So this is how this we can accelerate species discoveries. This is an excellent website, this waskweb.org, but not of India. It is of Afrotropical Hymenoptera. There also you can see interactive keys. You just have to follow that different options. You compare with the specimen, you reach in accurate identification. Such things is the need of the hour, but I should say, I have tried it out. It is time consuming, but we can do it. Taxonomy data, for what all are being used. It's not just to keep it as a dictionary of different species, but it is widely used as a baseline data for conservation oriented biomonitoring. Climate change is a very recent environmental threat, invasive alien species, also another biodiversity threat. Then for environmental impact assessment studies, for status surveys, all we need list of species. So the contribution of taxonomy to the larger society, to the citizen science, is by contributing field guides and the digital media, which you can now see in different websites. So these are all contribution by taxonomists. So we have a long way to go. Research efforts are not proportionate. Certain groups like charismatic groups, birds and mammals in the case of vertebrates, butterflies or donates in the case of invertebrates, large number of these works are going on and have already worked out. Whereas if you tell about insects, in the butterflies, Papilionidae, in the beetle, Lucanidae, Cicindelidae, tiger beetles and all, they are very beautiful to see. So people are largely after that to study. But other groups, many groups of beetles, then parasitic Hymenoptera, flies, all these are really ignored, and taxonomy is lagging behind in such groups. Another thing, when we compare the diversity of these two particular superfamily, I hope you remember when we explained that graph and all, these are the groups with largest diversity in India, Chalcidoidea and Ichneumonidae. Global diversity versus diversity in India. Globally, if you say Ichneumonidae is the largest group, but in India, if you say Chalcidoidea is the largest group. This is because mainly more workers were there in Chalcidoidea. So the taxonomic strength, the work involved was very high compared to this particular group, and that is how this diversity is showing more. So all this have relevance. Scientific relevance of published figures on species diversity. See, there is a latitudinal gradient in species richness. It is a persistent feature of the life on Earth. Towards the equator, you have more number of species, and when you go towards the pole, you have less number of species. Suppose this is an example from a termite episode in China. They were surprised to see a greater number of genus or taxa in higher latitudes. Why? Then the answer came, high taxonomic activity locally, just like we mentioned about the Chalcidoidea, that parasitic Hymenoptera in India. Many people were working and a lot of publications came, and that is how that more number even in the higher latitudes. Maybe an isolated biological process can happen, or it can be an artifact by taxonomic splitting, means some people are unnecessarily creating species. So all this can also happen. So when doing comparison analysis, again caution should be there on the species diversity figures. It may not be reflecting the actual diversity, but because of any of these reasons. Species have no political boundaries, so also is taxonomy. We need international collaborations. There is a great mismatch between biodiversity-rich tropical countries, and the experts are mostly in places where there is no much diversity. So when you have to study a species, we have to know about the variations. Suppose it is an international museum, you have the collections from different parts of the world, you get to know different character states of the species, and you will reach in better identification. So if you have some collaborators in other countries, they can also be a part of your study, and the overall output is refined and improved. But free exchange of specimens is not happening these days after the CBD and the Nagoya this protocol. There is some confusion regarding that genetic resources and fair and equitable sharing of benefits arising their utilization. See, exchanging specimens of taxonomy actually won't come into this category, but basic science started to suffer after this regulation came. Though sometimes permissions are obtained, it is too much administrative and laborious, cumbersome process for such permissions, and we are not permitted to send biological material outside India. And this is not the case of India alone. I am not quoting anything on India here, though the situations are very much similar. This is based on this particular paper, which explains the current situation in many countries like Brazil and Colombia. There are myths, there are facts. One great myth is that taxonomy is something you can do in your free time. Wrong. It is the biggest myth, I should say. Preparation of taxonomic publications, that is the most refined contribution of taxonomy, which comes in the shape of monograph, revisions, species checklist, that need lifelong expertise in the field and highly time consuming. It cannot be done as a part-time job. It is a full-time job, and taxonomy is not an outdated science. Many advanced things are happening. Even I can say the time some 25 years ago when I started taxonomy, how it was all line diagrams only. Now it replaced by digital images, by SEM images. Then the molecular taxonomy has come. So a lot of progress, and even the computer, the digital technology has changed the way it looks, the availability of literature, everything has changed. So it is also a very dynamic, not a static science. Another problem this is a fact, can be considered as a taxonomic impediment. Very less number of positions available for people to practice taxonomy, even after when they are qualified. The curatorial positions, they are burdened with other works, technical works, administrative works, supervisory works. They hardly get time to sit before their specimens, preventing them from doing taxonomy on a regular basis. A very sad situation. And the government policy in general shift from basic to applied research, strongly focusing on revenue generation. See, taxonomy is a basic science. Never it had an aim for revenue generation. So there is a shift for this granting of budget towards projects which generate money. And this is all from this particular paper, Challenges to Contemporary Taxonomy. To summarize everything, I could have gone into detail, but time limit is there. So regular updation of your identification tools, which are the keys and this taxonomic monographs or whatever, it should be regularly updated to accommodate new discoveries and changes in classifications. Then DNA barcoding or shape recognition, like the Google image recognition, all these things are there. They all need fundamental taxonomy groundwork. It is the next stage. Once the fundamental taxonomy morphological thing is worked out, only then the DNA barcoding or the shape recognition, all this can happen. Taxonomic framework has to be integrative with molecular data, behavioral data, ecological data, distributional data also, and all in agreement with morphological data. For the time being, we cannot do away with morphological characterization. That is the current situation in taxonomy in any group. So I hope I could cover almost the basic important aspects of exploring the unknown and the joy and the challenges, everything involved in it. Thank you. And this is the photo of one of the very exciting expedition which I could be a part of. This is a photo from Galathea, the Great Nicobar Island, which is the southernmost tip of India. Thank you.
Dr. Geloth.
Now, any questions?
Thank you so much. Yeah, thank you so much, Dr. Geloth, for the interesting and engaging talk. And we have got a couple of questions from our participants. So if you're willing to answer, then we can ask them.
Yeah, can I exit from this? Yeah, okay.
Yes. Okay. So the first question is, Aitgani Das wants to know how bees, ants, and termites develop the social behavior uniquely from other insects.
See, the social behavior seen in bees, ants, and termites are not alike. They are different. So, how it developed, I am not able to answer. Can go to the next question.
Yeah, sure. So the next question is from Sanjana Banerjee. Is there a need for
Yeah.
reclassification as a result of habitat destruction and organism redistribution?
I don't think so. A reclassification is not needed. Only you can identify based on the existing information keys or whatever the taxa. Classification is not needed. Reclassification is not needed.
Okay. And the next question is from Pravat Kumar Swain. He wants to know what is the nature of soil used for termite. Is it normal soil or different?
It is normal soil only. It is normal soil. Different kinds of soil are there, and different kinds of termites inhabit different kinds of soils.
And next question is from Krishnamurti S. Can you throw some light on how you capture insects for analysis and identification?
I think I have already covered that in the presentation. I have shown you different methods how to collect insects and what are the different steps once you collect insects. Also, you go for the preservation, then you go for either pinning, either mounting, and for the barcode studies, you have to preserve in 100% alcohol. I think I have covered it.
And the next question is from Sanchari Gurai. Is DNA barcoding done in classical taxonomy for identification?
No, this is done as a supplementary tool. For classical taxonomy, it's a standalone. It is an independent concept, and DNA barcode is a recently developed attribute. It adds that part. It is not a compulsory that DNA barcoding should be there to describe a species.
Okay. And another question from Sanchari is how much is the method of studying the organism phenotypically accepted only by observing under microscope at present time?
It is accepted 100%. Because phenotypes are not something different from the gene, means it is the expression of the genotype. So if you are an experienced person, you come to know which are vital characteristics and which are basic and should not be given that much weightage.
And another question from Hitagni Das is there any difference between systematics and gamma taxonomy?
Yeah, definitely. Gamma taxonomy is the population studies involved, and the systematics means you are even the phylogeny, everything is coming under systematics. The relationship between organisms.
And another one from Prabhat. International Code of Zoological Nomenclature. Can you give some examples of such codes?
I can't understand the question. In for naming, we have only ICZN here. International Code of Botanical Nomenclature is there. That is, the rules are different. I think for bacteria, for fungal taxonomy, and all different codes are there.
Okay. So with that, we are done with the question and Q&A
Yeah.
session. And
Oh,
Okay, so thank you so much, Dr. Keloth, for taking out your time to join us to deliver today's talk. It was such engaging talk, and you provided us with new insights on how taxonomy is important and relevant to our
Yes.
society.
Yes.
So I,
Thank you
I
for
hope I hope
staying with
future
me.
work will provide more information or like about how taxonomy can be very much helpful in our society.
Yeah.
Thank you so much.
Thank you. Thank you very much, and thank you for patient listening. I hope I could ignite some interest in the youngsters.
Yeah,
Thank
and
you.
that is very much important. Thank you. Thank you so much.
Thank you.
And to all the participants, thank you so much for joining and for all the questions. Sorry if we have missed some questions because of we were running late today. And do like, share, and subscribe to our YouTube channel. And our Facebook page also gets regularly updated, and we also broadcast it live on our Facebook channel. So do join us for the talks in the next week. And till then, take care and bye.